Hello, college biology. Here is Osmosis video, and this will probably take multiple videos. So, Osmosis video part 1. We'll see how far we get. Osmosis is the diffusion of water across a semi-permeable membrane. Well, as we know from diffusion, that means particles will be moving from an area of high concentration to a low concentration, and a semi-permeable membrane, or selectively permeable membrane, just means that particles will be moving through a barrier, and that barrier will let some particles through, but not all particles through. So some can pass through, but not everything. Now, this sounds a lot like diffusion. That's what's happening in diffusion. We've got oxygen and carbon dioxide molecules passing through the cell membrane, but it can't be just as straightforward because water is polar. If water tries to pass through that cell membrane, it'll get attracted to the phosphate heads. Water's got these charges. The phosphates have charges, so if we try to pass through there, the water will say, nope, I don't care about these non-polar fatty acid tails. No, I want to be attracted to these. So if we have a tunnel that has some charges in it, there's nothing that pulls the water back. So as it gets knocked through, it can just stay through and it'll pass through that tunnel. So for osmosis to work, we need a tunnel like this, and our example of that is aquaporin. Aquaporin is a transport protein that is required for water to pass through the cell. Otherwise, it would be attracted to the phosphate heads. But we are going to diffuse that water from areas of high concentration to low concentration. And to predict how the water is going to move, there is an idea called tonicity. Tonicity describes the concentration of solute outside the cell. And because diffusion has to do with high concentration to low concentration, we've got to know about concentrations to describe how the water is moving. We've got three circumstances here, three tonicities: isotonic solution, a hypertonic solution, and a hypotonic solution. Well, in an isotonic solution, the concentration of solute outside the cell is the same as the concentration of solute inside the cell. That also means that the concentration of water outside the cell is the same as the concentration of water outside the cell. This means we go back to the diffusion situation because we are at equilibrium, so an equal number of particles are moving into the cell and an equal number of water particles are moving out of the cell. The cell's fine. No big deal. However, we have other circumstances. In a hypertonic solution, hyper meaning greater, there is a greater concentration of solute outside of the cell. If there's a greater concentration of solute outside of the cell, there is a lower concentration of water outside of the cell. Since diffusion occurs in the direction of high concentration to low concentration, the water molecules will move from the higher concentration in the cell to the lower concentration outside of the cell. This will occur until equilibrium is reached, and if equilibrium can't be reached, then the cell will dehydrate and potentially die. Our third example of tonicity is a hypotonic solution. In a hypotonic solution, we have lower concentration of water outside of the cell, and that means that we have a higher, I misspoke. We have a lower concentration of solute outside of the cell. That means we have a higher concentration of water outside of the cell. And since diffusion is movement from high to low, the higher concentration of water outside of the cell will go inside the cell. Now, this will continue until equilibrium is reached, and if equilibrium can't be reached, then the cell will burst. And we call that lysis. If you have a rupture in the membrane, that is lysis. So that's what's going on with our cell membrane. Now, there's another idea, and that is called osmoregulation. And in osmoregulation, that is the regulation of osmosis to prevent dehydration and to prevent lysis. Sometimes the cell is in an environment that is going to be hostile, and the cell's got to do things to keep itself alive. It's got to maintain homeostasis. And osmoregulation is what the cell does that. We're going to look at examples of osmoregulation in the next video. So for now, let's review. We have osmosis, the diffusion of water across a selectively permeable membrane, where selectively permeable means some particles pass through and others do not. This requires aquaporin, which is a transport protein. Without this, the polar water molecules would be attracted to the phosphate heads of the membrane and they would not pass through. To determine the direction that water is moving, we have to use tonicity. Tonicity describes the concentration of solute outside of the cell, and we have three examples. In a hypertonic solution, there is a greater concentration of solute outside of the cell, which makes it a lower concentration of water outside of the cell, and so water will leave the cell to the low concentration until the cell dehydrates or equilibrium is reached. Isotonic solutions have the same solute and water concentrations inside and outside of the cell, And so the same amount of water enters and leaves the cell because the cell is at equilibrium with its environment. Hypotonic solutions have a lower concentration of solute outside of the cell, which means they have a higher concentration of water outside of the cell, so water will move into the cell until equilibrium is reached or lysis occurs. The membrane is ruptured. And finally, osmoregulation is the regulation of osmosis to prevent lysis or dehydration, and we have several examples which we will address in the next video. I hope this helps. Struggle well.